It is Tuesday, July 28th, and you are watching the Biotech Voyager. It's the show that covers what's next in biotech. We're mapping the early stage signals that
It's been a while, Ben.
Yeah, it has been. I'm clearly out of I should have done my audio exercises before starting. It is July 28th. It's a Tuesday, and you're watching the Biotech Voyager. We're covering the early stage signals before they become big news. Thank you for spending some time with us, and we are looking forward to the conversations today. We're going to be having a special guest at 11:30, about half an hour into the show today, from Deck Bio, Jack Silberstein, focusing on some really cool applications of T-cell engagers. Jeff's second favorite topic, probably. So looking forward to that.
Behind in vivo CAR T, obviously.
Yes, that is Jeff's favorite topic that we'll get to in a point. Excellent. But as always, where we're going to start the show today is to just quickly go on to the biotechvoyager.com and look at the leaderboard of the top early stage companies. Again, basically, we're ingesting all the different signals across the early stage biotech space. We assign all of those different signals a score, and then basically the companies with the highest scores get to the top. And the goal really is, we're not interested in, huge pharma acquisitions. What's far more interesting is, what are the early stage players that are going to get acquired 2 years from now, and you learn about them before anyone else does. And so this has been a super handy tool in terms of just really identifying those early stage companies. So Jeff, right off the get-go, Core Biomedicine. I know we're going to spend a little bit more time.
We're going to spend some time with them later. Yeah.
But they have jumped right to the top. What's the big signal there? And we'll get into it more later.
Series A. Utopia and Elecon. So I think Core is like a Boston-based company, but yeah. Congratulations. $21 million Series A, led by Utopia Innovation Platform and Elecon Ventures. So the we'll get into more of what this is later, but they're doing like pretty interesting cancer therapeutics, looking at like cell lineage and stuff like that, because cancer is ultimately a defect of like differentiation, kind of,
right?
Sort of. So they're looking at things like transcriptomics and using AI to develop precision therapeutics for oncology. And they just closed their Series A. And $21 million for Series A is like the perfect little range, right? We've seen some wild mega bust, like $200 million Series A's, and we're just like, what is going on there? So this makes more sense. Really cool. Small molecule stuff. Pretty sweet. Claris, they're doing ocular biologics. The big thing here, let's see, Series B, to fund their pivotal trials. Ebel Zeta, they've been on here for a while. You can see the various like steps happening here, lots of signals. Corbis, we'll go through some of these today. But then the big one here, Prime Medicine IPO'd, I think like last week, maybe.
Yeah.
July 23rd. Nope.
We had taken off last week. So we're catching some of these signals a bit late. But yes, Prime Medicine, big deal there.
They had like the pricing of their IPO was last week, I thought.
You might be thinking of Scribe Therapeutics.
I am thinking of Scribe Therapeutics.
Yeah. Which we will get to right after we go through this leaderboard.
So what did Prime Medicine do?
10, have they?
IND clearance. They haven't, but it was kind of a while. Oh, there it is.
There it is.
They're like super active. This is Jennifer Doudna, right?
Yes. Yeah.
Yeah. There it is.
And Benjamin Oakes, which is really funny because there's the Benjamin Oakes who's the CEO of Scribe Therapeutics, and I also know a Benjamin Oakes who lives in like Oshawa up in
Okay.
Canada.
Not the same person.
Not the same person. They
Mhm.
do look a little bit similar.
Mhm. If you Google Jeff Martin, you get like some Canadian singer. Not me.
Nice.
Definitely not musically inclined. But then I'm excited to go into this company later, Transcripta Bio. You know, Ben, I've told you a million times that we're gonna basically see. I think like basically like every therapeutics company is gonna have an internal like AI discovery thing.
Yes.
Basically every company that I picked today has some kind of like internal AI transcriptomic based cell or therapeutic discovery platform for oncology. So buckle up for those because you're gonna get about 3 or 4 of them. But
Well,
yeah,
that is really interesting, right? The and this is something that we've chatted about before, right? That there are all sorts of different platforms that are now coming out in terms of being able to spin out therapeutic candidates, and it's almost to some extent where the platforms, and I want to be careful in the way that I say this, to some extent they're almost like commoditized. Everybody's got one, which
yeah,
doesn't make them super special. What's going to be really interesting to follow up on in the next couple of years is who can successfully
yeah,
make candidates that get into the clinic and get approval.
yeah,
Because yeah, you've lowered the bar. It's kind of like developing an app to some extent, right? It's now
exactly that. Yeah,
super easy to develop an app. It's super easy to develop a novel molecule. The challenge is now adoption and effectiveness inside of the clinical landscape.
yeah. When it comes to like building your own SaaS, right? Like 10 years ago, it was you needed a team of engineers and da 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 da, and then it would cost millions of dollars to make these programs. And now you can make them in your basement, right? And the bottleneck in like that world is marketing. Essentially, the bottleneck you're going to see in therapeutics is like clinical data, which is tough because it's significantly harder to get clinical data based validation than marketing based validation.
Absolutely.
But there is that like perfect parallel. You're exactly right.
Excellent. Well, again, thank you everyone for coming today. We're going to be having a great conversation with Jack Silberstein, the CEO of Deca Bio, at 11:30. Stick around for that. If at any point,
Jack
as we're
from
going through these
Deck.
different stories, you have any questions, feel free to stick them in the chat and join us in that conversation. All right. So
Yes.
that being said, Jeff, should we focus on Scribe Therapeutics?
I did it again. Look at this, dude. My brain is just like, it's called Prime. Okay, here we go. What do you got, Ben?
All right. Well, I mean, the big news here, right?
Yeah. Yeah.
They
Huge.
have IPO'd for
Mhm.
a pretty significant amount of money. They came in at I think around $15 per share by the sounds of it. Again, a little bit of context for those of you who may not be as familiar with Scribe Therapeutics. We've talked about them several different times on the show. We think they're really cool. Spun out of Jennifer Doudna's lab, run by a guy called Benjamin Oakes, who randomly I also know a guy called Benjamin Oakes up here in Oshawa. In Canada, but basically they've raised $120 million in equity plus $75 million upfront from Lilly in a Prevail partnership, and they've got some really cool partnerships with Sanofi and Biogen, valued at over $3 billion in potential milestones. So a
Oh, is
lot
that all?
of different things going on with this company that are super
Yeah.
cool. But interestingly, when you think of Jennifer Doudna, you think of direct editing of
Yeah.
the DNA, right?
Yeah.
Cutting, editing, copy pasting, that sort of thing. Scribe Therapeutics takes a slightly different approach, where they are using the same kind of technology, right? You're still using cases and different enzymes in that regard, but it's more on the epigenetic side, where they have developed some really cool applications for essentially silencing the genes without actually going in and cutting anything out or knocking anything in. Instead, it's very much focused on what is the software over top of the genetic code that we can influence in one way or another.
Yeah, they can modulate the transcription of these things, right? And
Yeah.
I still don't really understand the benefits of that over like antisense oligonucleotide, like some kind of long non-coding RNA to degrade the mRNA or whatever. Both platforms would be regulating the transcription, basically, like the amount of mRNA available to be made into protein.
Right.
But
Well,
Scribe's approach just seems kind of more complex. The only thing that I can think of is like it's for transcripts, quote unquote, that are difficult to develop ASOs against.
could be.
And you could just kind of like target any genomic locus.
that again, the other thing I think is going to be interesting, right? Is just the reversible nature of it. If there is some sort of small molecule or whatnot that you can put in at a later date to reverse it if need be, that might be the advantage over a hard edit inside of your DNA.
Oh, of course, but but ASOs are also reversible because they're transient, right?
Right.
So like the question isn't what is the benefit over a permanent edit and this epigenetic modification. It's what's the difference between epigenetic modifications and like mRNA degradation, therapeutically speaking, right? I still don't really know, other than we have total access to the whole genome with like Prime's approach or Scribe's approach. Gosh, next time just like slap me in the head. Versus like the ASO space might be limited to like, can we design an ASO against this specific transcript? I'm not a super big like oligo guy, so I'm not super sure the limitations there, but it would have to be a direct response to some kind of limitation to oligos, right? That's got to be what it is.
I kind
So
of see I see oligos to some extent though, Jeff, as a little bit too far the other way, right? Editing is permanent for the most part. Oligos are transient. This is something that kind of melds the
yeah,
best of both to some extent, right? You've got something
fine.
that's going to last past a couple weeks. You've got something that could be potentially reversible, and it's a lot easier to do. It's
Fine.
kind of
Yep.
somewhat similar to, I don't know, the difference between uh, invisible ink versus writing with pen, and then this is a pencil that you could erase fairly easily.
Sure. Or like I think of like Lafabra, where they have this cell-based enzyme replacement that's not permanent, but it lasts a lot longer than like an enzyme infusion that you got to get every 2 weeks. This could be,
Exactly.
you know, maybe an ASO is like too transient, right?
But
And it
anyway,
just costs
if
more
anyone
for
over at Scribe Therapeutics is listening, we would love to have you on and ask these questions in person.
that would be insane.
Shout out to Ben Oaks.
Okay, who is next?
All right. So
I
coming
think
up,
it's code.
this is now core
Core.
biomedicine.
Core. Yes. Okay. So first off, Signal again, $21 million Series A from Utokyo and Elicon. And what this company is doing is actually really cool. It's a cancer therapeutic company, and I think it's small molecules. But this does a pretty good job. It goes beyond genomics. It's actually kind of a good way to put it, to target core biological pathways defining cancer cell identity. And this is the key word here, identity, right? And it's kind of a concept that not many people appreciate unless you're like deep in the weeds of cancer biology and like cancer genetics. Cancer tends to epigenetically a lot of times hijack like developmental programs, and they can sort of kind of become very stem-like.
Right.
And like, you know, obviously, Ben, you under the idea of like iPSCs, right? We can take a cell, a terminally differentiated like skin cell, and we can induce that thing to become a pluripotent stem cell, right?
Right.
Which means that every cell in our body has the potential to become any cell in our body. Cancer can, you know, not deliberately, but they do it. They hijack those kinds of transcriptional programs and can like reverse and push the cell down certain differentiation pathways towards advantageous proliferation, essentially, right? And the thing is that different kinds of cancer, based on genetic mutations or environmental toxins or whatever, can have different effects on like pathological cell lineage.
Yes.
So determining like an actual set of targetable things is super complicated. So you need what?
Core biomedicine.
You need AI. That's always the answer, Ben. Come on, you know this. So if you look at their website, although it's a lot of reading, but they essentially have, of course, you need to have this. This is what I was trying to get at. You need a combination of like this data science to get omics data, RNA sequencing, methylation data. This is actually probably super important in terms of like epigenetic remodeling, because there's really like two major kinds of epigenetic modifications. There's DNA methylation and histone modifications, right? And the easiest one to do is either ATAC-seq to get the chromatin accessibility, and then methylation to get like DNA methylation, both of which are heavily involved in like cell lineage. So I'm not super sure exactly what they're doing. They're being kind of close to the belt with what they're doing specifically, but I can basically figure out from this chart that they're getting a bunch of data from cancer cells using publicly available sources, like like the CCLE, and probably like proprietary data sets, using all kinds of AI on a big, massive data sets to determine like, hey, here's these specific targetable pathways that lead to transcriptional changes that lead to these erroneous or pathological cell lineage fates that you see in cancer. Really, really cool. I'm not super sure how well it will work though, because one of the key uh, uh, characteristics of this whole area of biology is plasticity,
Right.
right? It's kind of like punching a water balloon versus popping it. Inhibiting a pathway here in this space, I would anticipate you're probably going to get a lot of like compensatory reactions molecularly.
This is such a fundamental challenge with biology in so many ways, right? And I've kind of come across it throughout my career. I remember early on, right, I was working for Biotech, and they're focusing on antiviral defense pathways
Mhm.
to improve the production capabilities of cell lines for virus and that kind of thing.
Okay.
Obviously, cells normally don't like to make virus, and one of the huge challenges, right, is if you knock out or inhibit one pathway, it just kind of melds around it. The inner workings of our biology is so elegant and redundant in terms of making sure that the core functions continue to go forward.
Sure.
Like, sorry, I used
Elegant and redundant is normally not used in the same sentence. It's
They're-
elegantly redundant.
yes. And so it's really interesting to see this kind of playing out to some extent as well with therapeutic development as well. You've got to be really careful to ensure that whatever process you're going after or whatever pathway you're targeting, that there isn't a really easy way for your biology to basically get around it, and you're kind of sitting with an asset that kind of sits dead.
Yeah, and cancer is essentially pathological evolution, which makes the matters even worse. Like the cancer is good at one thing, not dying. Right? So
Right.
it's essentially just like, you're gonna have there's like there's the active compensatory reactions, which is like literally it can respond in real time to the drug. But then there's also the subpopulation of inherently resistant clones that will
Right.
just, whatever, outgrow once everything else is dead. So I suspect, watch, they're still preclinical, but watch this data. I would not be surprised if the only successful mechanism going forward is like combination therapeutic strategies. Because I don't suspect that this is going to work on its own. We'll see though. Who am I? Who am I to say, Ben? I'm an idiot. Who knows?
Okay, so but that being said, obviously they've gotten a $21 million Series A. Well done.
Mhm.
And then what is this AMED grant as well?
Oh. Core Biomedicine Japan received an AMED grant to support R&D and university partnerships. I don't know. That sounds like some kind of Asian thing. Let's see. AMED grant. Japanese Agency for Medical Research and Development.
Okay, that makes sense. Cool.
Yeah,
So this
cool.
company is based in Japan, and they've got a nice tranche of government funding to help them run through this thing.
They must have multiple offices because it even says like Core Biomedicine Japan, as in like there's multiple sites.
Yeah.
But still, yeah, pretty cool.
No, that is a common tactic amongst early stage biotech companies that have the capabilities to do so. Set up
Yeah.
shop in a couple different countries, and by doing so, you can harvest some of those different grant opportunities
Mhm.
to help you advance your research.
And here's so this is a little bit of like Sherlock Holmes esque take here. Why would you need to have a university partnership and like some kind of governmental sponsored grant for this kind of company? One of the things that I'm gonna harp on going forward in like biomedical R&D is the development and curation of proprietary data sets. And who is really good at developing proprietary data sets? Scientists.
Yeah.
In academia. I think that the university system is going to be a really powerful source for biotech companies to get a lot of like hands-on dirty work done to produce proprietary data sets, things like this. Like, we need to figure out what are the transcriptional characteristics of various cell lines, tumors, blah, 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 before, after treatment. We just need a bunch of like RNA sequencing and ATAC sequencing and single cell sequencing done. Who better to do that than an army of postdocs, right?
Right.
So let's get some government grants, university partnerships, have like lab and biotech collaborations. Love to see it.
So this is a little bit of a tangential question, but would that to some extent be a way that universities continue to maintain competitiveness? Because something that we've talked about before, right, Jeff, is
Sure.
there is this challenge for universities going forward, just kind of given the moat of knowledge that AI is filled in.
Mhm.
Does it make sense maybe for some of these different earlier stage people to not necessarily go through the university system and basically strike out at their own with some sort of incubator or some sort of like early stage investment fund? And
Yeah, I don't know.
realistically, right, the
Yeah.
challenge, the interesting place that universities would be able to come in the case of companies like Core Biomedicine, right, is you are the place where you can provide this brand new data set that's just going to be absolutely essential for any sort of company going forward when it comes to AI and developing therapeutics using AI.
Yeah. We could see, I suspect to see more things like that in the future too, for sure. Yeah.
Cool. All right. So yeah, this company is based in Japan. What else is there?
Well, they're not based in Japan. They're apparently, so the Voyager says they're based in Boston because because isn't everybody based in Boston? I'm pretty sure it's just like, oh, not sure where it is, probably Boston or San Francisco. Just like throws them in there. Yeah, I was just kind of looking at the various the thing is like my sources are mainly like European and North American press release sources, so we might miss a lot of the Asian market, but a lot of the Asian companies also will publish things in like the Western media outlets as well. So yeah, South Korea, Japan, booming.
Nice. There's not a whole lot of therapeutic development in North Korea.
Shockingly, no. In fact, there is literally zero.
Who would have thought?
Or
We definitely
Russia,
need to
for
do a
that
little bit
matter.
more work on building out China as well. But again, that's a bit more challenging
Yeah,
in some circumstances. Cool.
yeah.
All right. Perfect. All right. So we have one more signal we're just going to cover quickly before our interview with Jack Silverstein,
Jack.
rather, the CEO of Deck Bio. Really looking forward to that conversation on T-cell engagers, something that Jeff very much loves. He was very excited that we were able to get Jack on the show, so stick around for that. T-cell engagers don't get nearly the extent of love that perhaps they should. Really interesting component of science.
They have some issues. We'll talk to Jack about that.
Nice. Okay. And I imagine he's got these are questions that he's gotten before. So
it'll be a prime opportunity for the tell Jeff how wrong and dumb he is segment of the show.
a fan favorite.
Yeah.
Cool. All right. So yes, Liberate Bio, focusing on CAR macrophage therapies. Now,
how dare you, CAR monocyte?
I believe they're going after monocytes and macrophages. I could be wrong. If you click
What's
in on
the difference?
What is the difference, Jeff? What's the difference between a monocyte and a macrophage?
I won't bore you with my immunology.
Yes, but anyway, it is an interesting approach. Macrophages kind of went through a little bit of a like a hype period where they were really interesting inside of the solid tumor space, right? Because it's kind of dealing with some of the issues that CAR T therapies have faced historically, which is they don't do super well inside of the solid tumor. And the reason they're not doing super well inside of the solid tumor is partly because macrophages are inhibiting them and causing all sorts of issues. And so along comes kind of like the whole macrophage side of things where it's like, well, let's just kind of flip this on its head and let's kind of use the enemy against itself. And so that's kind of the underlying concept. But as we were discussing just before the show, Jeff, right? The challenge with macrophages just over the last couple of years has been like some high profile failures. Doesn't mean that this approach can't work. In fact, that's why we wanted to include Liberate Bio, but Charisma for therapeutics, for example.
Yeah, Charisma was like the biggest, CAR. And to answer your question, macrophages are monocytes. Uh,
There we go.
Basically, any myeloid cell, like neutrophils, dendritic cells, macrophages, are monocytes, kind of. So, but yeah, there was a big hype around macrophages, and the idea isn't necessarily to like take the enemy and turn it against itself on the tumor. Like we can reprogram the macrophages. Companies who are doing those kinds of things are companies like Feast Therapeutics. Feast has like uh, macrophage targeting antibody therapeutics, kind of. They do don't eat me blockades,
Right.
right? Which is a way to activate macrophages. There you go, and destroy tumor cells. The idea of like using a macrophage or a monocyte cell therapy is to actually leverage the therapeutic functionality of the macrophage themselves,
Right.
right? And macrophages are good at, let's say, three things: getting into tumors. Okay. They love just like hanging out there. And there's a whole thing that I can go into about how tumors are kind of like unhealed wounds, and macrophages and neutrophils are like really good at getting into wounds and like eating up bacteria and stuff. So macrophages and neutrophils are usually the first cell types or the most abundant cell types within solid tumors because they go to the site of the wound. And then once they're there, they're good at that, getting in there. They're also very good at eating stuff and secreting stuff. Okay.
Right.
The problem in tumors is that they're also, I guess the fourth thing is they're also very good at like being plastic and changing based on like what they've eaten, right? And it's good in wound healing because you want them to be very inflammatory at first to bring in immune cells, to bring in certain cells to heal the wound. And then you want them to polarize and be very anti-inflammatory to allow the wound to heal and the homeostasis to return, right?
Right.
So that the polarizable nature of macrophages is actually very good for our body, but it's not good in cancer because the tumor just gets kind of stuck in this we're healing a wound phase, and macrophages get stuck there. But they're actually really, really good at like killing cancer cells and eating them and secreting pro-inflammatory stuff if you can keep them there. So the idea with a macrophage-based therapy is let's make these things stay like M1 macrophages, anti-tumor, pro-inflammatory macrophages, and just send them in and make them eat the tumor. But the interesting thing here is that this is a CD19 targeting CAR monocyte platform, which is not really meant for solid tumors. It's for heme, like B cells.
Right.
So I don't know, man. I don't know.
Yeah. No, it seems like an interesting company. Just kind of keep in mind they've developed out a Raptor platform as well. They've kind of gone through they've got a therapeutic component that's entering the clinic. Yep, they're going after autoimmune disease.
Okay, so it isn't even oncology.
Yeah,
it's like immunology. Okay, that
yeah.
makes, I guess, more sense. But it says cancers. Yeah. Okay. Well, doesn't matter.
Yeah,
it's cool regardless. That's a really cool cell type. They're just touchy and they're hard to manufacture. So we'll see. Good luck to them. Charisma had I call it like a massive failure. It was tough. They had some negative results last year. And it's a big bet, but we'll see how it turns out for them.
Excellent. Well, Jack Silberstein has entered the green room, so we'll be bringing him on in just a minute. But yeah, I guess just quickly, Jeff, again, what was really interesting just for Liberate Bio's approach, again, is the fact that they are using LNPs along with, and I thought this was interesting, they are also developing their own platform for this. So they're approaching the clinic, they're going after autoimmune disease, and approaching first in humans. So we want to cheer on a truly novel approach to just kind of see if this kind of thing works. We've tried a bunch of different approaches so far with regards to autoimmune applications, and it'll be cool to see if this works as well.
Okay, so it actually isn't even a gene therapy, or it isn't a cell therapy. It's a gene therapy. It's like it's in vivo CAR-M.
That's correct.
Okay, very similar to Create.
Yes.
In fact, I'm pretty sure that Create. Yeah.
Anyways, you're a fan of them.
Yeah, they're doing like a whole plethora of like T, NK, and myeloid.
Yes.
Yeah, really cool stuff.
Excellent. Cool. Well, Jeff, that being said, Jack has very kindly given us his time. It is just about 11:30, so we're going to get started here. But yeah, should we bring him on the show?
Mm-hmm.
Excellent.
Do it.
All right, perfect. Well, ladies and gentlemen, without any further ado, let's bring him on.
Jew.
Jack, welcome to the Biotech Voyager.
Hey, Ben. Hey, Jeff. It's great to meet you. Thanks for having me on.
Oh,
You
we
as well.
appreciate your time. Jeff's a big fan.
Glad to hear it.
Yeah. I'm a big fan of just like really cool biology. So just, whatever. Love T cells, love T cell engagers. And this is, I don't want to jump into it too quickly, but this is the first of its kind therapeutic. I haven't seen anybody doing bispecific peptide loaded MHC antigen T cell engagers. So looking forward to talking to you.
Yeah, we're trying to pave the way here to
Yeah.
get things out there.
Yeah, for sure.
Right on. Well, excellent. Folks, if you have any questions for Jack, feel free to put those in the comments and we'll get to those post-haste. But I guess just in terms of like an intro here, Jack, can you give us a little bit of your background? Where'd you come in? What was kind of the idea for starting the company start?
For sure. Yeah, no, can go back to kind of the beginning of, my training. I previously was a clinical research coordinator at Johns Hopkins, and that was enrolling phase 1 to phase 3 studies, mainly focused around metastatic prostate cancer. And it was mainly biomarker selected trials. And during that time, got really familiar with clinical development, then wanted to learn how to, make drugs, early side of things, and then went to PhD at Stanford for training as a protein engineer. And then what we're doing at Deck Bio is really trying to combine these two sides of things in terms of, clinically informed protein engineering, where with single targeting approaches, we haven't really seen that compelling of data. There's some early inklings of data coming out now, but really trying to use this multi-targeting approach to really stack, the efficacy signal, stack response rate, stack durability, and lead to, greater overall survival for patients. And so we were started in September of 2023 and have raised just under $4 million to date to deliver this novel medicine, specifically for our first program targeting HLA-A0201 with these 4 different targets.
Fantastic. So if we back up for a second here, right? In terms of the problem that you guys are going after, what's the key indication?
In terms of the indication, the target indications, or in terms of why we're going after this type of approach?
Well, we'll back up for a second, just like the disease, I guess.
So right now we're focused on lung cancer and then gastroesophageal cancers. We also have head and neck cancer as our priority indication as well.
Gotcha. Okay. So
oh gosh, I don't want. This is gonna be so tough for me, Ben. I gotta like just like give you your space, but I have so many questions.
No.
So esophageal cancer you mentioned.
So both types of esophageal, both adeno and squamous, and then also stomach cancer as well, gastric cancer.
I'm going to guess that came from the bottom up. Like you guys had antigens in mind and it wasn't a disease thing. How did that start? You don't see a whole lot of esophageal cancer therapeutics other than different surgeries, basically.
So basically it was more the other way around. It was mainly looking at clinical data to say, okay, what is necessary in order for patients to be able to respond to these types of T cell engagers? It's completely different game than ADCs, where you can have a very dirty target and very great efficacy and safety. With T cell engagers, you need something that's completely clean in terms of healthy tissue profile. And then the other thing is, if somebody doesn't express the target if they have target 0 levels of expression, they're not going to respond to therapy. If they have some level of expression, there's a chance that they can respond. But really, when you look at the clinical data, it's patients that have high levels of expression that are the ones that are most likely to respond to therapy. We've seen this, at least in the PMHC, TC side of things, with both Immunocore and Immatics. What Immunocore does is they measure protein in the tumors and have shown that patients that have high levels of expression are the most responsive. With Immatics, they measure mRNA as a surrogate for peptide MHC. And they've also shown that patients that have no expression don't respond. Patients that have high levels of expression are more likely to respond. And so we have looked across the entire proteome and looked for targets that are expressed by greater than 30% of patients at more than this response threshold that we've identified. And so really focused on trying to drive, greater response rates with these types of medicines. And, individually, these targets, not one of them is the standout. It's really through the combination that we're able to have much higher expression levels at both an mRNA, a protein, and a peptide level to drive more durable responses.
So, okay, then is there a therapeutic advantage to doing bispecific PMHC versus like one PMHC, one surface antigen, maybe kind of a mixed bag? Is there, or was it a design choice?
So the way that we identify these targets again is mainly looking through clinical data and then combining that with RNA, protein, and peptide databases to confirm that these are actually expressed at high enough levels. And then the key enabling part for us is also that at the surface level, when these are presented as peptide MHCs, that the peptides are structurally similar enough, even though they're not the same at a sequence level, that at a structural level, that they're similar enough that with one T cell engager, you're able to recognize these multiple peptide MHCs. And so it's, a simple kind of 1+1 bispecific for our lead program. And then in terms of thinking about, traditional cell surface targets, when we were starting, there was no FDA approval for any type of cell surface target. Now we have DLL3 as a target that's been approved. But when looking across, gastroesophageal cancer, lung cancer, all of these types of different indications that we're focused on, there's no clearly compelling cell surface target for a T cell engager approach. There's a lot of antibody drug conjugate type of approaches out there against, dirtier targets.
Sure.
But in terms of clean targets, the majority of them are expressed inside the cell. And so that's why we're focused on peptide MHC as a target class to allow us to access those.
And that's I recently learned about DLL3 as a target. It's so cool. It's basically like a spillover effect that only ever happens in cancer, right? You've got something that just gets so rapidly produced inside of the body, inside of the cell normally, and then it just kind of in specifically cancer, so much of it gets expressed, it basically just blows out to the surface. It's a really elegant marker to go after, so that's nifty.
It's one of these oncofetal kind of embryonic, targets where it's expressed during development, but then silenced in adulthood. And one thing that's interesting about, this T cell engager versus ADC field, with ADCs, you're going after, HER2 that has 1 million copies per cell. With DLL3, it's about 2,000 copies per cell on patient samples, like, logs lower. But with something like a potent T cell engager, you're able to address that versus, traditionally with ADCs, with old school ADCs with MMAE, the thought was 10,000 copies.
Yeah.
The cutoff. Anything below that you couldn't hit. And then when you're talking about peptide MHC targets, you're another log below that when you're thinking about tens of copies or hundreds of copies.
Yeah, there's like 2 on there. But the thing is that the T cell engagers actually engage the full like T3 complex, even versus like a CAR. A CAR is less. You need to have a higher surface expression of a CAR target because they just don't function as robustly, like in terms of the T cell activation. So you mentioned, I want to go back one second. You mentioned that the targets are structurally similar enough that they can both be recognized by the same T cell engager. Do you mean that either arm of the bispecific can bind to either target because they're that similar?
So the bispecific one, in our case, when you think about a classical T cell engager bispecific, it's that simple Y shape, where one arm is binding to the tumor and then the other arm is binding to CD3 on T cells. And so it's really through this one arm that we've done a significant amount of engineering to get this very precise recognition of multiple cancer-specific targets.
Okay.
Not recognizing the other, 10,000 plus healthy targets.
That's some real nerdy stuff right there. That's really cool.
very, very deep structural and
for sure.
Which was my PhD at Stanford.
Okay, so I was thinking more of like a tri-armed structure where you have like target 1, target 2, CD3. But you're saying it's like it's a standard like Y-shaped mAb, but the one arm is just like hyper intelligent, basically.
Exactly, highly engineered, human-like in the sense that we, expect, lower levels of immunogenicity, but then
Hmm.
are very engineered in terms of its stability, its developability, and then, most importantly, its specificity. And that's, another reason that we focused on using T cell receptors, which are the natural protein that recognizes peptide MHC complexes. A lot of people have tried to develop antibodies
Right.
that recognize peptide MHC. They're called TCR mimetic antibodies. We've seen a lot of issues with those in the field in terms of their specificity. And so we focused on maximizing the specificity with a T cell receptor. And then we've engineered it in such a way that it's stable and looks like an antibody in terms of its manufacturing profile.
Yeah, I feel like a variable region is just too clunky to do like a 15 peptide or 15 amino acid peptide recognition, right? You need to have like the canonical biology in there.
And when you look at peptide MHC structures, the majority of them are in this kind of canonical docking where it's recognizing across the entire peptide MHC. With an antibody, it could be
right. It's just like, this looks cool.
Recognizing.
Yeah.
So it's a little bit more random in terms of its specificity.
And is Immunocore mimetic, or are they a true TCR?
They're a TCR.
Yeah.
They have a very different approach in terms of going after single targets and some additional pieces that are different.
Yeah. But it's good validation because they're approved.
Yes, they've paved the way. But again, really low response rate, durable overall survival benefits. But we're really focused on having something that can, through this stacking effect, have, actual responses.
Yeah.
For them, you're talking about single digit percentage response rates.
Yeah.
Awesome. Okay, so if we back up for just a second here, right, when we're thinking about, you're going after gastroesophageal cancer and whatnot, why have other approaches to these particular diseases failed? Why is there an opening here to use T cell engagers? Why have other approaches failed?
For sure. I would say, first off, in the case of T cell engagers, there's been a lot of failures. In terms of gastroesophageal cancers, there are exciting, data coming out now where, earlier stage treatment with chemo immunotherapy is looking really promising, especially in the neoadjuvant setting, pre-surgery. When looking at something like esophageal squamous cell carcinoma, very few therapies that have trodden that path. I think it's about a 6-month overall survival in the second line.
That sucks.
And so, you just, there's a lot of just when you think about cancer as a definition, it's just very heterogeneous.
Yeah.
And, there's a lot of exciting ADCs coming out. But, one of the downsides of, ADC, which is just, chemo slapped onto an antibody, is that, it's only working as long as you're on it usually, versus something like an immunotherapy. The goal is, you can treat for a certain period of time if the tumor is, shrunk enough that, you could stop therapy and the patient's immune system could still keep the cancer in check. And so, when thinking about these types of cancers and why they might have failed for certain indications, it could be that the targets that are out there are not particularly compelling, where they have healthy tissue expression, so there's no therapeutic window. Could be that, for the developer, the sponsor, the pharma, that they didn't do type of a biomarker selected trial, so they weren't able to enrich for those responders. And now people are going back and going from an all-comers approach to a narrowed set of patients.
Yeah.
And so I think that there's a lot of exciting things coming up, and we're excited to bring a T cell engager type of approach to these types of patients.
Yeah, those carcinogen, like the cancers that are mediated more by like carcinogenic exposure, things like lung cancer, esophageal cancer, GI tract cancer, they're in melanoma, they're more amenable, I think, to immunotherapy because of the variability. Whereas more like genetically predisposed tumor types may respond better to targeted therapy because they're more predictable.
For sure. And one thing that's exciting though is that we are seeing, dramatic responses in these traditionally cold tumors with T cell engagers. With checkpoint inhibitors, it requires that there's that endogenous recognition.
Yeah.
With a T cell engager, you're creating an artificial immune recognition. And so we've seen, beautiful responses in prostate cancer, where something like STEAP1 PSMA. I mean, PSMA has a troubled past.
Sure.
But STEAP1 and KLK2 both showing great activity with T-cell engager approaches.
So I have a very nerdy question that I think is interesting. Hopefully you think it's interesting too. Would there be some kind of benefit to knowing the intratumoral expression of the subsets of T cells that already are expressing the TCRs that recognize those targets? Because I feel like if you know the target's there and there are T cells that are there that have the proper TCR and they're not responding, your therapeutic probably wouldn't respond that well.
Not necessarily, because for a T-cell engager, all we require is target expression. So for something like a cell therapy, there's all of the, additional, like, is the T-cell happy? Is it, getting the right co-stim factors, et cetera? For us, all we require is the peptide MHC is being expressed on the cell surface at a high enough level of expression. And then it might be that we don't even hit those with the other arm, the CD3 arm. We might not even hit those specific T cells that are dysfunctional. We might hit a completely different, non-TCR. It might be a influenza reactive TCR, T cell. And so there's no endogenous specificity required. It's just, whatever T cell is available and ready to, do the job.
Interesting. So if we back up for just a second here with regards to the fact that this disease that you're going after is very much underserved, in terms of even the other approaches not doing well with regards to this. Like, what was the early data that you saw with T-cell engagers where you're like, oh shoot, we've got something really cool here?
Yeah. So right around the time that we started the company, September of 2023, first ever T-cell engager for a solid tumor indication was approved in 2022. So T-cell engagers have been around for a long time, have a lot of activity in blood cancers. Now we're seeing activity in autoimmune indications. In terms of solid tumors, it's very nascent. And we had the first approval in 2022 with Kimmtrak from Immunocore. And then in 2024, we had the second approval with Imdelltra tarlatamab for DLL3 and small cell lung cancer.
Is that Amgen?
That's Amgen. Yep.
Yeah.
And so that was, as we were getting, up and going, that was showing really great clinical activity. And, DLL3, though, it's not really expressed in many tumor types beyond small cell lung cancer. And then that's a very small fraction of lung cancer. So we were looking at, how do we address the larger fraction of lung cancer? 85% of lung cancer is non-small cell lung cancer.
Yeah.
And so the thought was, look across the entire proteome, the entire genome, to identify, these peptides that look similar enough that you could have this stacking effect to have, much higher levels of expression. One target that a lot of people are going after, that it's a single peptide MHC targeting approach, is a target called PRAME. Yeah. It's like the CD19 of the peptide MHC space. Yeah. Everybody
and MAGE, right?
Everybody in our space all has a PRAME targeted program,
or KRAS G12V.
Definitely up and
TCRs. Yeah.
But showing greater potency than a PRAME molecule, and now with this PRAME clinical comparator that's out there, it's showing really strong activity in melanoma and ovarian cancer, where one patient with ovarian cancer had a complete metabolic response, and the first gen of PRAME targeting was developed by Immunocore. Now there's this next gen approach from Immatics, where with half-life extension, in terms of a T-cell engager that has much longer half-life. With Immunocore's TCs, it's about 7 to 8 hour half-life. With this new gen, it's greater than a week. We show greater activity than
Sorry, just a second. So the standard currently for T-cell engagers is about 7-8 hours, and you guys are at a week.
Yeah, exactly.
Wow.
So just trying to make it a lot more convenient for patients, because with Kimmtrak, you're getting a dose and it's on a weekly schedule. So you get a dose, you might have it in your system for that day, and then you have 6 days where you don't have any drug, treatment. And so with these next gen approaches, we're seeing much greater responses. And we're looking to build off of that with multi-targeting.
So what's driving that? Why is the expression, or why is the durability that much longer?
Pegylation.
So what Immunocore does, they were the leader in the field. They were paved the way, basically. They've been around for a long time. They developed TCRs that have a stabilizing mutation such that you can manufacture them. But then one of the issues is that it has such poor stability, the molecules that they have, that they require expression in bacterial systems, where most biologics that are being developed today, and most antibody-like molecules, are not expressed in bacterial systems.
Yeah,
they're expressed in CHO, which is a mammalian type of system. And so what we've done is we've added on an FC domain, the FC of an antibody that provides that half-life extension, and then enabling us to express in a CHO-like system. And then in terms of the TCR that's really causing a lot of the developability, manufacturing issues for them, we've created this proprietary technology for stabilizing them such that they look like an antibody when going into a CHO system. So it's just a very simple purification schema, and then very high purity for dosing.
So do you guys, with an FC competent antibody structure, is there a potential for like fratricide, or I guess the destruction of like FC receptor positive immune cells?
That's a great question. So what most people do, we're not novel to this, but most people make certain mutations in the FC that you knock out binding to FC gamma receptors. You still bind to the neonatal FC receptor such that you have that half-life extension, but then you don't have any type of binding to complement to FC gamma receptor. No binding to macrophages or anything.
Yeah, in case. Yeah.
Exactly.
That's cool. I want to learn more about the ARM, but I don't want to get too big into the Stanford protein engineering history. But that seems like it's some kind of like alien, with it's got to have some kind of crazy end on it, I'm going to guess.
It's surprisingly simple. It's mainly just structure-driven, protein engineering. And so,
really cool, man.
It's really, taking advantage. TCRs, they naturally have some low level of cross-reactivity.
Yeah.
We've just done this in a site-selective manner to create that selective, multi-peptide MHC recognition, and then really ensuring we built this platform where it's called DB Scope, where in a sequence agnostic manner, we've taken everything that is expressed in healthy tissues, trying to make sure that we don't bind to any of those. So we have these 4 cancer-specific targets where they have completely clean, healthy tissue expression profiles. There's no level of real protein or RNA expression of those targets in healthy tissues. But then making sure that we don't cross-react with a healthy peptide MHC. We for every molecule that we make, we experimentally screen against every potential cross-reactivity in terms of healthy peptide MHCs.
Yeah.
Mitigate any type of, risk that we've seen with other types of TCs.
Yeah. And how easy would it be to expand beyond HLA-A2 or AO2?
So our second program is actually a different HLA, where our first program is focused on HLA-A0201, which is the most common HLA in people in the US and Europe. For our second program, it's going after HLA-A2402, which is the most common in people of Asian descent. About 60% of the Japanese population expresses that HLA.
Yeah.
And it's still taking advantage of the same concept of hitting multiple peptides, but then different peptides, completely distinct peptides, but from the similar protein group, then being presented by a different HLA.
Okay. So it isn't just like the same peptides on different HLA. It's a different thing completely, basically.
Correct.
Yeah. So wow. That's complicated.
A lot of more protein engineering fun.
Yeah. Yeah.
Nice. So I guess just to in 30 seconds or so, what would you say is like the one piece of data that you've seen so far that is just really compelling when you're talking with investors or different groups that would be interested in seeing your science move forward?
So when looking across being able to look across many patients, there's a lot of data sets that are RNA-based, single-cell RNA-seq, protein-based. For all of these data sets, we blow the competition out of the water in terms of, it's clear that this is, very compelling in terms of target prevalence. The key thing for peptide MHC is actually confirming that the peptide is expressed at a mass spec level. And so with RNA and with RNA-seq, it's very inexpensive, relatively expensive, but in the general world, but in the biotech world, it's very inexpensive to do single cell RNA-seq. With doing mass spec based approaches, it's multiple logs more expensive.
Yeah.
And so having that compelling data to show, okay, compared to what existing targets are out there, that we have significantly higher levels of expression than these other types of approaches, single targeting approaches, that we expect to have a more compelling response profile as a result of that. We've also shown preclinically that we have a more compelling response profile. I think those two things combined with our developability package, showing that we have something that's an easy to manufacture, simple in terms of it, dosing for patients. I think all of those things combined are really the key differentiation for DEC Bio versus other molecules out there.
So the standard, like I like how you express that, where it's basically like we're effectively increasing the expression of the target antigen.
Exactly, and again, we've seen at RNA and protein levels in clinic, from clinical samples that are for patients that are being treated with these therapies, that again, high levels of expression at those levels, at RNA or protein, correlates with response rates. And then the key thing that investors also want to see is, okay, at the peptide MHC copy level, that you actually also have higher peptide MHC copies as well.
Mm-hmm.
And everybody has their different flavor of what they think is the minimum threshold of copies per cell that is required for a T-cell engager approach versus a CAR-T approach versus a TCR-T approach. And every pharma also has their own different internal thresholds. And so, I think that it's very molecule dependent. It's very target dependent. It's very, cell line dependent. If you're going after a cell line that is not going to respond to a type of immune engager type of approach, whether that's a CAR-T or TCE, there's just a lot of variability between data sets on this.
Very good. Awesome. I guess there's two questions we always like to end on. First of all, what's next for you guys? What's next for the company? What should we be watching for over the coming months?
For sure. So the next thing for us is really narrowing in on what's our final drug that we're going to take into manufacturing. And I'd say we're narrowed down to 3, potentially 2 at this point. And our goal is by the end of this year to narrow down to just 1. And then take that into manufacturing. And then manufacturing is the biggest timeline driver for these types of biologics. In order to make it in this mammalian cell system, you have to make a stable cell line and then have it produced at high enough quality and purity that you can dose it into a patient. So then that's about 12-month timeline, depending on what vendor you talk to, it's about 12-month on average, I would say. And then starting clinical trials after that.
Very good. Fantastic. What's the dose for a T-cell engager? Like, how much are you putting inside of a patient?
Well, with the half-life of a week, probably way lower than you would need to normally.
So with Kimtrack, it's in the micrograms. It's it's like when the pharmacy is preparing it, there's like multi-step dilution. You have to be very careful with handling it, that you're not dosing the patient too much.
Yeah.
Because these things are so potent again.
Yeah.
And that's going after a target called GP100, which does have healthy tissue expression at low levels throughout the body. With this half-life extended PRAME molecule that's showing data now. Because of the cleanliness of PRAME as a target, it doesn't have very much expression in healthy tissues. they've been able to dose up to 30 milligrams in terms of what they've publicly disclosed. They've also said that they're dosing up to 45 milligrams.
Oh my God.
And so it seems like,
just like, take more here, have some more of that.
and they've actually shown that higher dose levels correlates with better response.
Of course, yeah, yes. Okay.
And so, you know, if you have a clean target, you can dose up to those higher levels. If you don't have a clean target, it's really challenging to push past, you know, what we've seen with, you know, Amgen has these multiple programs, you know, DLL3 being one. They also had a PSMA. They also have the STEAP1 in partnership with Xencor. If you have a clean target like DLL3, they were able to dose up to 100 milligrams per patient during the clinical trials. But now, you know, the FDA approved dose is 10 milligrams. But if you have a dirty target like PSMA, they couldn't even dose above a milligram. It was just too difficult because of the therapeutic window. So having a clean target allows you to dose higher. And what we've seen is that higher dose and then also higher expression of the target in terms of copy numbers are really the key two pieces in terms of driving more durable responses.
Fantastic. Excellent. All right. I know we're just about at time here, and Jack, we really appreciate your time and want to be super respectful of it. There is one final question that we try to ask all of our guests. Your answer can be a conglomeration of everything that we've discussed so far, or it can be something entirely different. And the question is just, what's slept on in biotech right now that's going to be a big deal in a couple of years?
I would say, you know, just as a continuation of our conversation in our field specifically, it's really specificity engineering and thinking about, you know, thinking deeply about, you know, making sure that you're not hitting any of these healthy peptide MHC targets. I think there's a lot of molecules that, you know, people are rushing into the clinic that are more like these TCR mimetic antibodies that I think are going to have some challenges with finding that therapeutic window because, you know, you're hitting some other type of healthy tissue. In terms of what I'm interested outside of, and I don't think it's slept on. I think in vivo CAR T is looking really exciting these days.
I gotta go, Ben.
You know, a lot of
Jeff has a love-hate relationship with in vivo CAR T.
I could see it coming. I knew you were going to say it.
I think it's so dependent on what's the you know, modality, whether that's, you know, lenti, adeno, or you know, LNP type approaches. But I think and whether you're targeting CAR T versus, you know, CAR M or anything else, but you know, really exciting data at ASCO this year. So excited to follow that along. I'm more cheering them on from the sidelines than doing anything active there, but yeah, really exciting.
You know, I have seen some taking the in vivo approach and the T-cell engager approach and kind of putting them together. Do you know the company Vironexus?
I don't.
So what they have is a LNP, or it's an AAV delivered, I think, T-cell engager construct. And they have like hepatocytes express it. Okay. And it just kind of secretes it into the blood as a T-cell engager. I think it's for like CD19. That's pretty cool, I thought.
There's been a lot of these types of in vivo production of proteins. I think like, you know, again, CHO is so established. It's, you know, you know exactly the dose of molecule that's going in. When you're doing any type of in vivo expression, it gets me a little scared in terms of, you know, your hepatocytes might be really great and producing tons of this molecule. Mine, you know, might be a little, you know, more crapped out.
Right.
And so I think
too much drinking in college, huh?
Exactly. I think it's just too much variability. I think, like, you know, if I'm the FDA, I'm a little bit concerned about that. But I think the, you know, T-cell engagers, we know them. We the field has matured significantly. The FDA has gotten a lot more comfortable with them from the initial, you know, scary experiences in the early days. Now we know so much about, you know, dosing strategies and mitigating cytokine release syndrome. Yeah. And so there's just a lot of, you know, learnings, and now it feels like we're finally maturing as a field.
Fantastic.
Agreed. I like it.
Excellent. Jack, thank you so much for your time. We really appreciate it. Awesome. Thank you all. Really good connecting. And excellent. All right. We'll see you later. Cheers. Awesome. That's a super nifty approach, Jeff.
Yeah, I hadn't, the fact that it's one single arm, like targeting arm, is super crazy. That's actually like really awesome. heavily like engineering expertise required for that one. And the idea of just kind of thinking of it like we are in effect increasing the expression of the target molecule to hit that threshold and get it to work. Yeah, really, really nice. So
kudos to Jack.
Looking forward to seeing their clinical data.
Yeah, that's awesome. All right, excellent. So Jeff, estrogenics, I believe, is the next one here. You want to take a look at it quickly?
Yeah, I'm trying to figure out, what are we looking at here? Is this my here we go. Perfect. Estrogenics. They got a little bit of seed money recently.
Yes.
$2 million seed series by Talent Funds. Yeah, Ben, what are they doing? I mean, estrogenics is probably doing estrogen something, right?
Yes. So this one's cool. Because again, it's a focus on women's health, obviously, half of the population. And in particular, they are going after the whole menopause space. Widely kind of like an untapped market that doesn't get a whole lot of attention. But it's like huge unmet need. And so basically what they've been able to focus on here is an approach that's going to be going after this. So it's a women's health play, focusing on an unmet market need that has
this is not where I thought it was going to go. Yeah. Normally when I think of like ER stuff, is it an antagonist or an agonist?
I'm not sure.
Because typically, okay, it's an agonist. That's different. ER antagonists are like breast cancer.
Right.
So I thought it was going to be a breast cancer therapeutic company. Oh, was I wrong? They reduce hot flashes.
Yeah.
And improve memory. Does estrogen must have some kind of effect on the brain? I mean, duh, what the heck? I'm stupid. Of course it does. And it's post-menopause. See, this is like totally out of my wheelhouse. Menopause biology, I have no idea. Right?
It's a fascinating area that doesn't get a whole lot of attention the way that it should.
But I'm wondering, like, can't you take like just like hormone replacement or hormone like guys take steroids, right? This is like the same kind of thing, but for like postmenopausal estrogen reduction.
I think it gets a bit more complicated, and again, I am by no means an expert in here, just kind of given the whole reproduction system that women possess for making babies. That plays a larger role than, just taking, like for guys, right? Replacing testosterone might kind of allow you to do some sort of components of longevity, but it gets a whole lot different when you've got like a reproduction system that's also kind of like, as it were, shutting down.
Well, it's also, so this is, but here's where I was thinking, like, this is not new, right? Like taking estrogen is not new.
Yes.
But taking estrogen that bypasses the cancer and cardiovascular risks tied to ER alpha targeting hormone therapies, that is new. That's new.
Okay.
This seems to be like the play. We have it's a new kind of ER beta agonist that's going to bypass, because if you just pump somebody full of estrogen, they're going to get like breast cancer, ovarian cancer, like,
okay.
So
this is really the interesting play then. The fact that they have developed estrogen that allows you to take it in larger doses than you would normally so that you don't have that cancer risk.
Yeah, that's actually really interesting. And there must be some kind of like cognitive and like physiological effects of postmenopausal estrogen reduction. Duh. But then it would appear as though the classic canonical approach would just be like ER alpha agonists. And it's like great, but also like cancer and cardiovascular damage. But this is all the good with less of the bad. Seems like that's like the thought, at least.
Yeah. And they recently got a new CEO, I believe, as well.
Let's just go to their website and see what the deal is. Privately held pharmaceutical company. Blah 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 blah. Where's the team? Okay, yeah, Victoria.
Yes. So, fun fact for those of you who are listening to this, Victoria should be coming on the show either this week or next. So, looking forward to that. So, if you have more questions about this, feel free to come to that show. Details are not yet confirmed, but she will be coming.
Cool, very cool. Yeah, okay. again, I'm not super keen, or I guess I'm not super aware of like the nuanced biology of estrogen receptor therapeutics, but it seems as though like this is more of we're trying to avoid the potential negative effects of the classic estrogen approaches.
Gotcha.
Oh, it's Midwest too. Awesome. Are they based in Milwaukee? Nice. That's cool.
Let's go.
Yeah.
I think ASGCT is based in Milwaukee as well. Like
Really?
the actual
Huh,
organization,
like the hub?
American Society for Gene and Cell Therapy.
Hmm.
You wouldn't think it, but yes,
Yeah,
they
I
are.
think I would say Boston, San Francisco.
Yeah.
Cool.
Right on. Excellent. Okay. So that leads us on to the next signal, Jeff, I believe. But yeah, folks, stay tuned for that. It's Transcripta.
Ooh, okay, yeah. This is I forgot that I picked this. AI-powered transcriptomic platform for small molecule discovery across multiple diseases. Is there not cancer in here? Oh my goodness, it's not a cancer play.
Jeff,
Interesting.
Here you are thinking you've got an easy cancer play, and it's
yeah, I mean, you would think
more nuanced.
an AI-powered transcriptomic platform. I think immediately cancer. Duh, right? But no.
What else does do transcriptomics do?
Yeah, what else does biology do besides cause cancer? The conductor AI model predicts biological responses by matching drug signatures. Hold up. To the inverse of patient-derived disease signatures. Wait, what?
Wait, what? Come again?
It predicts biological responses by matching drug signatures to the inverse of patient-derived disease signatures. As in, this drug causes 1, and the disease state is -1. Let's put them together to get 0.
Interesting.
Lead programs target neurological and neuromuscular diseases, including Huntington's disease, myotonic dystrophy, FSHD, autism. Wow. Autism?
Good luck with that. that is
But that would make sense though. It actually does make sense that it's like neurological, because we can just say like, okay, transcriptionally, like transcriptomically, Alzheimer's people or people with autism tend to display phenotype A, and we know that this drug causes phenotype -A. So 1 - 1 = 0. That seems to be what this is, right? Let's just look into it. Let's just go to their website, because that's got to be what it is. We find that reverses. Yeah, that's super cool. A disease is a signature. We find the drug that reverses it, of course. I mean, it sounds simple. Like, why has nobody thought of that before? You probably couldn't do it. You need AI. Conditions where decades of drug development have come up short: neurodevelopmental, neurodegenerative, rare muscular diseases, each share complex transcriptomic dysregulation, true, that traditional approaches can't address.
Okay.
Signature reversal.
Realistically, if we're just pulling in from the whole like AI component, right? You've got vast data sets of transcriptomic data, and it's like a combination of like 17 different things, and being able to kind of use standard approaches in the past, it just wasn't going to be viable. With something like AI, you now have that ability to kind of look through all of that data and kind of pull out the non-intuitive insights, where it's like the combination of these 17 different factors is what causes the inverse of disease state.
I think it's simpler than that.
Yeah.
I think it's we know the transcriptional pattern of the disease, and we know the transcriptional effects of a drug, and we can just match those up. Like, the drug tends to inhibit this kind of we can just test that. Like, give cell line drug, test transcriptional changes, right? You get those transcriptional changes, either some positive, some negative, and you can say, okay, if this drug caused gene A to go up and gene B to go down, and in autism, gene A is down, gene B is up, we can just give that drug to fix that. That seems to be what it is.
Interesting.
Which is only possible once you have reliable and robust methods to acquire the data, RNA sequencing data, and analyze it with AI.
Right.
And then you can just do it's like a Tinder for drug development.
Nice.
Basically.
Super cool. Well, yeah. Transcripta. Well done to you guys. Whereabouts
what
are they
was
based?
the? Of course. Where else would this kind of company be? Palo Alto. And then what was the signal? They raised a Series A. They have money. They have money.
Yeah. And they
We
have the
should
Nano
talk to
Clinic
them.
on board, as would be indicated by the title that we've got on the screen here.
Where? Oh, nice. The one that you made? Yeah, really cool. Really cool. Where's the CSO? I wonder what like the scientific background of this is. I don't see it. This might be like oh, I'm not signed in here. Anyways, yeah, cool. Does it mention like where this came out of? I'd be very curious. Yep. Ben, do you know what this is?
Yeah.
Oh, never mind. This is just RNA sequencing. I thought it was a microarray. Never mind. This is just RNA sequencing data. Oh, it's single cell RNA sequencing data. My bad. Yeah, it's got to just be like we have a bunch of single cell sequencing data from patients, and we can get like a signature of a disease at the single cell RNA level. And then we have a bunch of single cell RNA sequencing from like drug experiments, and we can just kind of find the inverse.
In a way, isn't this almost a similar type format to what we saw with Alchemab? Alchemab is using AI to identify antibodies. These people are kind of going to find the inverse drug that's got like the inverse transcriptomic profile of a disease state.
I think that this is just my opinion. I think that Alchemabs is cooler because it's just like it's like a magical soup. It's like Alchemab is basically saying, "Hey, you seem to not get sick a lot. Can I have some of your blood, please?" That's Alchemab's approach
for science.
Right? Yes, it's for science. I promise. I'm not going to drink it. I promise. But I guess in effect we kind of do. It's like the whole plan, right? But this is more like investigationally robust, I guess you could say. Maybe. I don't know. It's similar vein for sure.
Yeah. And again, yeah, I think right. It speaks to something that we could do to an okay extent 5-10 years ago. And given all the advances in AI, it now just makes the whole process that much faster and becomes a bit more viable.
There you go. Look at this. This is exactly what it is. Okay, so you see here we have drug, or what is it? Yeah, so the blue is like the autism transcriptional like matrix with some kind of axes to define like to get this plot, right? And then the white is the drug. And you can just try and match up the peaks and the troughs to get like the inverse.
Nice.
Super cool. Yeah, that's really cool.
Fascinating.
Mm-hmm. I have no idea what the axes would be. That's part of their model. But yeah, that's how it works. Oh, you can move it. That's cool. Yeah, see how they're trying to match up those things like that?
Yeah.
It's like
essentially
make it symmetrical.
1 minus 1 equals 0.
Yep, precisely.
Super nifty. Cool.
Really cool.
All right, Jeff. There's one more signal for today. A company that we have spoken about before that I added in at the very end. And you are... And I just want to show this to you. Hiya. Do you remember them?
No.
No?
No. What happened? What is it?
Jeff, do you remember us talking about the dark genome?
Ooh. Of course I do. Of course.
Yes. So that's what. So for
cool.
longtime listeners to the show, several months ago, we discussed this company called Hiya Therapeutics. And in particular, what they had developed was a therapeutic approach that goes after the dark regulatory genome, basically the large components of the genome that are kind of like untranslated regions. In the past, they were kind of considered to be like junk DNA. Obviously, they are not. But basically, what they have been able to do is they take their approach to develop long non-coding RNAs to then go after diseases. Targeting these different like regulatory regions and whatnot. And so they are developing antisense oligonucleotides, Jeff. You were talking about this at the very beginning of the show.
Mm-hmm.
This is one such approach. So it's super interesting to see that the FDA is now basically seeing how cool this approach is that we were talking about a couple months ago, and have awarded it fast track designation.
Okay, hang on, because now I'm beginning to think more nuanced about this biology. Okay. It's Scribe? Scribe is doing the uh epigenetic
Correct.
CRISPR stuff.
Yes.
Okay. Yeah. So sometimes long non-coding RNAs, like pathogenic long non-coding RNAs, can actually be synthesized or be produced from a RNA mRNA processing event of a gene that is actually required and needed for normal cell growth and normal cell function. So you have like, here is good gene being made, boo, 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 and then during processing, long non-coding RNA is produced. And this is pathogenic, right? If you just suppressed the expression of this gene to remove the presence of this RNA, that would be toxic. So you could use a ASO, an oligonucleotide, to selectively degrade this while not affecting this. However, a more robust approach, if you could, would be to just stop the expression of the whole thing, right? Which is what Scribe does. This seems to be like it's more of, this is like a shooting it out of the sky versus stopping it from taking off.
Yeah. And anyway, the FDA has seen that this is something that's pretty cool and deserves some fast track designation.
I will say though, this is a little bit of a moot point, but using the word dark genome to describe like long non-coding RNAs is kind of cheating, because long non-coding RNAs, the term non-coding is in their name, right? So like the dark genome is like non-coding, right? It's like, yeah, of course, long non-coding RNAs are a part of the dark genome. That barely counts, okay? I'm talking about like things like stochastic expression of like random pieces of DNA that aren't just, although I guess it would be non-coding RNAs then, technically speaking, but whatever. This is just me being a nerd.
Yeah. But anyway,
Ben's like, yeah. You are being a nerd.
As one nerd to another. But anyway, their lead asset is an antisense oligonucleotide targeting WHISPR, a cardiac-enriched long non-coding RNA that drives fibrosis and heart failure, with pipeline programs extending to pulmonary fibrosis, obesity, and oncology. So, yeah, a play inside of the heart disease space.
Yeah. Yeah, really cool. I'm wondering, yeah, see, again, my brain just goes right to cancer. And again, there is a lot of plays in oncology, but like thinking of long non-coding RNAs in cardiac disease, it's just like, oh yeah, they definitely do things there too, I guess. But I learned about all these things through the lens of a cancer biologist. So it's like, right. So yeah, pretty cool. I mean, looking at it'd be like, what is the long non-coding RNA doing in cardiac muscle that's making it pathogenic? Right? Oh, it's affecting the contraction of the muscle. Oh yeah, that would definitely be a problem. Right?
Indeed. Excellent. Well, folks, we have to wrap up just a little bit early today, but thank you for coming. We were able to discuss quite a few different early stage signals, a lot of really cool stuff happening inside of early stage biotech right now. If you want more above and beyond what we have talked about here on the show, go check out the biotechvoyager.com. Feel free to check out, you can filter even by like indication or disease, stuff like that. You can filter by location by going to the map. Lots of really cool stuff over at the Biotech Voyager. So if you are in need of more signals, you can go check that out. Yeah, Jeff, you want to just do a quick little
I wonder if let's do this, right? This is just the leaderboard. So this is only companies who have signals, right? Why is Haya not on here? Dang it. I wonder why. Doesn't matter. But yeah, let's hang on. Do this. Cardiovascular and where's cancer? Oncology. Bing, bing. Sure. Alphabet. They're not here either. That stinks. Okay, whatever.
You have to go back and look at the tags that were used.
Yeah, sometimes it's like if there's so many different tags, then or like different like focuses of the company, the tags are like, what do I do?
So wait,
I can't put everything.
Just go back to Haya for a second here, because I actually want to see this. What are the tags that are being used for it? Cardiovascular.
There's not. It's RNA oligonucleotide, not gene therapy. Duh. It isn't a gene therapy. That's why. But it would have caught it if I wasn't stupid.
So if we go do that quick, if we go look at that leaderboard. Okay, so cardiology, for example, and RNA oligonucleotide.
It'll be number 1. Yeah, for sure.
Perfect. Right on. I didn't know they're based in Switzerland.
Well, now you do.
There we go. Right on. So yeah, folks, we are going to be shutting down the show in just a couple minutes here, but if you want more signals, go head over to the biotechvoyager.com and you can like filter. It's like a running live list of all the early stage companies that are super cool. We've talked about a bunch of them today, but if you want to specifically look at one particular indication or one particular modality, or you want to look at a particular indication within a modality, that is all capable over at the biotechvoyager.com. It's completely free, so check it out.
Completely free. Excellent. Forever and always.
Yes, indeed. Right on.
You're welcome.
Perfect. Yes. Excellent. All right, folks, we're gonna close it out now, but we really appreciate you coming, and we look forward to the next show. It's going to be on Thursday. We're going to be having a special guest. We're going to be having the founder of Artan Bio, a gentleman called Michael Torres, who sold his company to Eli Lilly for hundreds of millions of dollars. It was inside of the bispecific space, and now they are making a play for longevity gene therapies.
Yeah, with tRNAs.
Yes.
Perfect.
Yeah. Phenomenal. So feel free to stay tuned for that. We haven't put the event up yet, but we'll be putting it up soon. And we would love for you to join us and ask some questions. Michael Torres, has successfully exited a company to Eli Lilly for hundreds of millions of dollars, and now he's on to his next thing, which is longevity gene therapy. So we hope you can join us on Thursday, same time as always. All right. Thanks so much, everyone, and we'll see you next time.